The American Brain Tumor Association is pleased to welcome you back to our webinar series. Our webinar today will discuss the role of genetic information in understanding and treating low-grade glioma. My name is Andrea Garces, Program Manager at the American Brain Tumor Association. I'm delighted to introduce our speaker today, Elizabeth B. Claus, MD, Ph.D. Elizabeth B. Claus, MD, Ph.D., is Professor and Director of Medical Research in the Yale University School of Public Health, as well as attending neurosurgeon and Director of Stereotactic Radiosurgery within the Department of Neurosurgery at Brigham and Women's Hospital in Boston. She is a member of the Board of Advisors for the Acoustic Neuroma Association, as well as the Central Brain Tumor Registry of the United States, CBTRES. Dr. Claus's work is focused in cancer and genetic epidemiology, with an emphasis on the development of risk models for breast and brain tumors. In addition to her research activities, Dr. Claus trained as a neurosurgeon at Yale New Haven Hospital and completed a fellowship in neurosurgical oncology at Brigham and Women's Hospital. Her clinical focus is on the treatment of meningioma, glioma, acoustic neuroma, and brain metastases. In partnership with national patient brain tumor organizations, including the American Brain Tumor Association and the ANA, Dr. Claus is working to develop cost and time-efficient web-based recruitment strategies to be used in the study of brain tumors. Thank you so much for joining us, Dr. Claus. You may now begin your presentation.
Thank you, and good afternoon to everyone. Just listing my disclosures and support here, and wanted to thank the American Brain Tumor Association, who's helping us to present our work and also to get a start on our low-grade glioma registry. So I thought I would start just with a definition. People use the term low-grade or lower-grade glioma in a number of different ways, so I wanted to list what I'm using the term as. So essentially, low-grade glioma is a generally slow-growing tumor of the brain, and it's called a glioma because it arises from glial cells in the brain. So traditionally, and this is an important point that's moving very quickly in terms of this field, gliomas have been graded from 1 through 4 based on the World Health Organization histology groups. So although there is some variability, generally grade 1 tumors occur primarily in children, and pretty much are an entity separate from adult grade 2, 3, and 4 tumors. So the tumors that I will be discussing today are adult grade 2 tumors, and I'm using the term low-grade glioma. These include astrocytomas, oligoastrocytomas, which are also known as mixed gliomas, so you might see either of those two terms, and oligodendrogliomas. High grade, which are grade 3 and 4 gliomas, include anaplastic tumors as well as glioblastoma. So one piece of information that's important to note and that I'll discuss a little bit is this past summer there were 2 very exciting reports in the New England Journal of Medicine, one led by researchers at UCSF and Mayo, and a second by members of the Cancer Genome Atlas Group, or TCGA. And their analyses, which have been confirmed by other groups, suggest that tumor marker and genetic profile data likely are better predictors of outcome, prognosis, and potentially response to treatment than these traditional WHO histology subgroups. So it's actually quite a big change in terms of how we look at things, how we think about treating patients with low-grade glioma, and as well as the information that we give to patients. So just some numbers to start. These are taken from the Central Brain Tumor Registry of the United States, which does include data from the National Cancer Institute. And so this is a listing of all brain tumors from 2007 to 2012. So it includes both benign and malignant tumors. And when you go through the numbers, there's approximately 3,000 new low-grade glioma, and those are adult low-grade glioma tumors, diagnosed in the United States every year. The mean age when we did some statistics on the data set was about 41 years of age. Obviously, the range is from about 20 to 60. Higher rates are seen in males as well as in whites, and that's pretty consistent across country and across data set. So when I'm talking about risk factors that relate to glioma, we know that there is not much known in terms of environmental risk factors for glioma. And the two big ones that have been confirmed across a wide variety of data sets are exposure to ionizing radiation and then an inverse association with allergy. So then we look at things that are genetic. As in any cancer, inherited genetic predisposition occurs in a small percent, and pretty much whether it's breast cancer or brain cancer or colon, we think it's about 5 to 10% of cases have some sort of inherited genetic component. In part, we know this because individuals that have a first degree, and that would be a parent, a sibling, or a child that have a diagnosis of glioma, those individuals have up to a twofold or twice the risk of a person in the general population. We do know that there are rare genetic syndromes that we don't see terribly frequently, and they include glioma as one of the potential diagnoses that show up in these families. So you may have heard of some of these, Lee-Fromini, neurofibromatosis 1 and 2, but these families are quite rare, and they really explain only a small proportion of familial risk of glioma. It's actually quite rare that we run into any of these families within a general brain tumor clinic. Now, the one thing that is interesting and which has been recently discovered over the past, say, 6 or 7 years, is that there are additional, and the ones we know about at the moment, number 8, inherited variants associated with risk. I'm going to talk a little bit about those as well and whether there's any implication in terms of screening for both individuals or family members. So when we talk about genes, it's a complicated topic, but we're talking about two types of genes. So one is what's called inherited. Another term that you might see is germline. So these are the genes that are passed on from parent to child. And when we as investigators attempt to measure something about that, the way that we get the information is after a patient consents, we draw blood or we get saliva or we can use a cheek swab. And so essentially we're able to get from those materials genes that might be related to glioma. And in addition, there's a second type of gene that we talk about, and this is actually in the glioma tumor itself. Another term that is used is somatic, or sometimes people will talk about acquired. So there are things that are inherited that are in all the cells of your body, and then there are things that are in the tumor itself. So the findings recently have related to both of those things, but you have to just kind of keep straight in your head when we're talking about which one. So we're now learning that in glioma, both these type of genetic changes are important, both things that are in the germline and also things that are acquired and are in the tumor. And we know for some of these tumors, we think that these acquired changes might be related to exposure, for example, to radiation, for a lung cancer patient, to smoking. So the environment can be important in that as well. So when we talk about inherited genes, so these are the ones passed on from parent to child, there are some that are quite rare, but generally associated with a very high risk. And one that is very famous that I'm sure many patients have heard of is the breast cancer or BRCA1 and 2 genes. Rare in the general population, but if an individual has the gene, they have a high risk of breast, ovarian, and other cancers. Some of those sort of genes have been found glioma. In addition, polygenic are genes that are relatively common in the general population, but associated with a much lower risk. And those are the 8 that we mentioned, and we'll talk a little bit about. So what's the evidence that inherited genes exist for glioma? as I mentioned, and to just look at this column here, when we've done in some of our large epidemiology studies, and I just give the reference here, this is the Glioma Gene or the Glioma International Case Control Study, led by Dr. Melissa Bondi, who's now at Baylor. We have found that in general, if an individual has a first-degree family member affected with glioma, then their risk is twice that of the general population. So that gives us evidence that there are genes that are inherited that seem to relate to glioma. So how, as researchers, do we go about looking for these genes? And there's a lot of different ways, but there are two main ones. One is we try to identify families in which there are multiple individuals who have been diagnosed with a glioma. A second method, and these require thousands of people, is we look at groups of individuals who have glioma and individuals who do not have glioma, and try to compare genes or genetic variants across the two groups. So I know probably a number of you have participated in Glio Gene, and just to let people know, this is still ongoing. It's led, as I mentioned, by Melissa Bondi, who's a professor at the Baylor College of Medicine. But this is an international effort to try to identify families that have 2 or more living members with glioma. We've been able to identify over 500 families so far, and actually been able to identify in particular one gene that appears to be associated with both glioma and melanoma. Presenting here just a table of one family, so each line gives a generation of family members. In this particular family or pedigree, you can see that they have 6 individuals with glioma, and notice there's a few other types of tumors as well. So very rare, very difficult to locate, and we're hoping just to make people aware of this study in case they have any interest to help us find these families. And I have the website down here as well as the contact individual below. So as I mentioned, there have been some genes identified that are rare but have a high risk of cancer, one of which might be glioma. This POT1 is a new gene identified through the Glio Gene study, and these are some others that have also been associated with glioma risk. So what about the more common inherited genes? This was what I mentioned in terms of looking at groups of people that have glioma and comparing genes in persons without glioma. And so this requires thousands of study participants. In some of the studies that we've organized, we have over 10,000 individuals. So as you can imagine, very expensive, very time-consuming, but needed to find these rare genes of note. So in 2009, as I mentioned, 2 groups found evidence of such genes, and I have the references listed here. What was amazing, and this does not always happen in epidemiology, is each group independently found evidence for similar genes, and others have been able to verify that. So that very strongly indicates that these genes are of interest. So these are the 8 that I've mentioned that we've found so far. These are the formal names. This is what we think they do, and you can see the word unknown is listed a number of times, so we obviously have a lot to do in trying to figure out what these genes do or do not do. If you look over in the final column, you can see that some genes seem to be related to certain types of glioma. And for those of you interested in grade 2, you can see something called TeRT. Some other genes are noted to be specifically or more likely to be associated with grade 2. So one thing to note here, and I haven't put numbers in, but it's important to think about not only is a gene associated with risk, but how great is that risk? So you can have genes that are associated, but the risk is not very high. So for example, where you see a single plus sign, that's about a 20 to 40% increase in risk. You do note we've got one or two here that have a couple of positive signs. So this particular candidate gene has a 6-fold increase in risk in terms of a certain type of glioma, and this one here has a 2-fold increase in risk. So that's something as a patient or physician you think, well, gee, that seems like a pretty good increase in risk. Should I or should my family member be tested? So there's actually a very nice review of the topic, and I've listed the reference here, regarding whether or not people should be screened in the general population. And at least at this point, screening for any of these variants, even those with higher risk, is not recommended for the general population. And the thinking behind that is the general risk of these gliomas is about 0.1%. So even if the risk is increased, so now it's 0.6%, that's still quite a low risk. And the cost of putting people through MRIs or screening, and we're not even sure how we should screen people, and it's not clear that intervening helps people in terms of their prognosis. At this time, there's no recommendation for screening in the general population for any of these variants. And the example I have here to compare to is the breast cancer gene that we mentioned earlier. In that, in the general population, there's a risk of breast cancer, and this is among men and women, of about 13% over a lifetime. Individuals that carry the BRCA gene have up to a 65% risk of breast cancer over a lifetime. Therefore, screening is suggested, and that is in part because there is an intervention that has shown to offer benefit in terms of survival and outcome. So just to compare that to the breast cancer scenario. So what about the other types of genetics that we talked about? So not germline or inherited, but in the tumor itself. What do those tell us, and how might we use those to select our treatment, to think about outcome, to make general clinical decisions? So I will tell you at this point, we are all still learning. It is an imperfect science, but it certainly is intriguing, and we're starting to get pieces of information. So as I mentioned, in the New England Journal last summer, there was a very exciting article, one by the TCGA group and one by researchers at UCSF and Mayo, and that's the article I have presented here. So essentially, they had data from a number of large population-based studies, and so they were able to go back and compare when I have information on histology, on germline genetics, on the tumor, what pieces of information seem to be important. And it came down, despite some very sophisticated analyses and large numbers of tumors and patients, that there were 3 pieces of information, and these 3 pieces relate to tumor characteristics: a mutation in a gene called IDH, a mutation in a gene called TeRT, and deletions or missing pieces of arms or parts of 2 chromosomes, chromosome number 1 and chromosome number 19. And so what they found was that outcomes, clinical characteristics, what the genes looked like in the inherited component differed across each of these groups. So they came up with names for some of the larger subgroups. So triple positive means they have a finding or a mutation in IDH, TeRT, and they have co-deletion. Triple negative means they don't have any of those changes, and then there are some groups in between. So the 3 symbols here relate to 3 different data sets, and so the point of that is to show that their findings were consistent. They found it in the Mayo group, the UCSF group, and the TCGA. So that indicates that there's probably good evidence that what they're seeing is correct. And so what this shows is that there are certain age at onset characteristics of the different groups. The triple positive group seemed to be the youngest, and the TeRT mutation-only group seems to be the oldest. There is association with outcome, and this does not incorporate any clinical information, so that's important to note. This is not incorporating any effect of radiation or chemotherapy. It's simply, this is what the tumor showed, this is how individuals did. So the triple positive and the TeRT IDH mutation group did best. One thing to note, and this is true across many studies, is that the data are both grade 2 and grade 3. So that's one point we want to note, and why we're interested in further pursuing grade 2 is generally the grade 2s, because the number of patients is smaller, are lumped into higher grade studies. So we don't have great information specific to grade 2s in terms of how they respond, what prognosis is. So that's one of our interests in terms of both genetic and other factors, is focusing on just a grade 2 group to give patients a little bit better information about what they can expect with that diagnosis. So what are these markers? So essentially the IDH is a metabolic enzyme, and if there are mutations, it alters the cell's ability to program itself to divide and to act normally. TeRT is another gene, and it helps to control whether or not the chromosome continues to enlarge or shorten, and that relates to cell death. And then this is unclear in terms of what gene is associated with it, but we know that loss of this area on chromosome 1 and this area on chromosome 19 is important. So let me just go back to here. Again, the point in this slide, which is taken from the New England Journal paper, is the 2s are always joined with other grade tumors. They're rarely presented separately. So it's difficult to give good information to patients with a grade 2 because they're always mixed in with other groups. You do see, however, that there are certain characteristics of the lower, and that's 2 and 3 groups, that differ from the higher groups. So you can see there are many more triple positive than if you go to a grade 4. There are many IDH mutation individuals versus grade 4. So you can see the characteristics of what is seen in the tumor varies quite a bit by grade. So don't want to spend too much time on this. I've given the reference below. The point is, it is important to know both information about tumor genetics as well as inherited genetics in order to be able to tell patients what sort of outcome and response to treatment might you expect. So tumor tissue testing is ongoing. Most of the large neuro-oncology centers, I'm at Brigham and Women's and Dana-Farber, but also Mayo, UCSF, MD Anderson, Memorial Sloan Kettering. All of these centers are now starting to add increasing markers or genetic changes to the panels that are tested. And I will tell you, this changes constantly. I just had a patient who had been treated about 8 years ago, and that tumor was not tested at that time really for any of these changes, because even over that short period of time, we've learned a lot, and we know now that there are things we need to look at. So this is just an example that I made up, but it's based on the sort of information that we give to our patients, looking across different genes. So you can see here's TeRT. This is where it's located, and in this particular tumor, there's no change or no mutation detected. BRAF for particularly pediatric tumors, but for other tumors as well, is looked at, and here's the 1P and 19Q. So this is the sort of report that you would get, and that would help the neuro-oncologist to decide what sort of treatment and what sort of tumor it is. It helps us to better tell what the pathology is. So does treatment strategy vary by tumor genetics and markers? I will say in terms of low-grade glioma, we don't know a lot about this. we're gaining some information, and certainly there are some intriguing findings. But again, that's why we'd like to try to gain more information on grade 2 to give patients a little bit better information. So surgery at diagnosis, I think that in general, the consensus, at least among neurosurgeons, is that gross total resection is the goal. It's obviously important to pick a good neuro-oncology or neurosurgical center that has things like intraoperative MRI and mapping and other key surgical tools to help get the best outcome. There has been no clinical trial looking at extent of resection and outcome. However, there's clearly a strong correlation between extent of surgical resection and increased survival. It's not only important for that, but to get enough tissue to make sure that we know what the pathology is, what the grading is. Obviously, if you get a small piece, that might give you one piece of information that may not be a complete picture of what's going on in the tumor. It's also important, if eventual clinical trial entry is to be considered, that sufficient material is obtained and stored to allow a patient to be eligible for clinical trial should that become necessary in the future. Now, one thing that's quite interesting is there's some new data for higher grade glioma that suggests an association between IDH mutation and benefit from surgery, so that people might benefit more from surgery if they have certain mutations. And I've listed the reference right there. However, it's not a clear-cut picture. The reason is, and this is a little bit of a complicated slide, but tumor marker, and I have a graph here for the 1p19q change as well as for the IDH change, and location are correlated. So these are the general areas that we talk about in the brain. It turns out that the frontal lobe, which surgically is a bit safer than some of the other locations, is more likely to show IDH mutation and more likely to show 1p19q co-deletion. So all these factors are correlated and interrelated, and hence it's difficult to know if the survival benefit is due simply to having surgery in a place that is a bit more safe, or because the markers exist more frequently there. So lots of things that we need to continue to look at. So another important question is whether or not, should a patient have a recurrence, whether or not surgery should be undertaken, or whether we should assume that the tumor is the same as it was prior to any intervention. Some patients will have had treatment in between initial diagnosis and recurrence. Others will just be managed conservatively with serial MRIs. So in the past, I would say that it was generally not suggested that surgery be redone. And obviously there are many factors that go into this, how long the time has been in between initial diagnosis and recurrence. But we are now considering surgery more frequently, and that's because of information that is presented in this paper, again led by the UCSF group in Science of 2014, which followed a number of patients, and admittedly a small number, but followed 23 patients who had initial diagnoses of grade 2 astrocytoma mixed or oligodendroglioma. They then looked at time of recurrence, what the tumor looked like. And what was interesting is that in 43% of the patients, at least half of the mutations that were in the initial tumor were not found in the recurrent tumor. And so the message there is things can change in terms of mutations from start, and this is just a little tree diagram showing for all their patients how they started and what mutations or changes occurred over time. So it may be important at least to consider whether additional tissue might be needed at time of recurrence. So you can see here, this is one patient, initial surgery, recurrence, recurrence, and over time, the different mutations, some of which may be treatment related, that occurred over time, but to treat a person at each time point might require an updated piece of information in terms of mutation. So at least something to converse with your physician about. Chemotherapy, most of the clinical trials to date have included the higher grade individuals. Some of you may be aware that there's new data out that look at both higher grade as well as high risk, low grade patients. And now that survival time or time since start of clinic has increased, PCV, which is a type of chemotherapy, has been shown to have added benefit in terms of survival. But again, we need much more data for low-grade glioma. Radiotherapy, I think we know even less. There are hints that IDH1 mutant tumors may show increased sensitivity to radiotherapy, but we really don't have much trial data to base that decision upon. So that leads me into our goal to get more data for low-grade glioma. As we mentioned, the low-grade glioma patients generally only a convenient subsample in studies of higher grade glioma. And so then when you're drawing results or conclusions from these studies, it's going to be driven by the much larger number of high-grade glioma. So we've now formed a consortium. The consortium is called LowGlio, and it's UCSF, Yale, Brigham and Women's Hospital, Mayo, a number of other individuals. And so we met this past summer, and across all the studies and sites, at least within the United States, we found only 651 low-grade glioma cases that had the sort of data available to allow us to address some of the questions that we've brought up in the talk. So I think that points out that larger numbers of low-grade glioma cases are needed that have all the information, so clinical, tumor, germline, so that we can focus separately on these individuals. And I think we're also interested in hearing what sorts of quality of life or symptoms patients are having, how can we get information to patients, and how might we make things better for people. So we're partnering with this LowGlio group to create the International Low-Grade Glioma Registry. We're trying to use patient organizations, and as I mentioned, the American Brain Tumor Association has started us on our way by giving us some funds to start development of our web-based registry. We're also going to create an iPhone app, and hopefully eventually for folks that don't have an iPhone, an Android, to try to make sort of a cost-efficient way, and so that everybody gets access to it, means to collect data for low-grade glioma. So we're hoping that enrollment will start this summer, and we're working with a lot of great patient organizations, American Brain Tumor Association, National Brain Tumor Society, the International Brain Tumor Alliance, and other websites. As I said, we're going to have patients enter via internet study website. We're starting with iPhone, and if that goes well, we're going to work our way over to Android. And so we are asking, and this is a part of what helps to make it cost efficient, we're asking patients to help in terms of the management of this study. So we'll be asking patients to identify or locate their pathology report and to get that to us, and you can mail it or fax it or email it or scan it, whatever is good for you. We have ways of uploading it on your phone or your iPad or other device to get it to us. So we do that to make sure that we have a uniform study group, that people that we say we're studying actually have the diagnosis that we think they have. And so I just wanted, these are some of the poor, hardworking graduate students and project director, Dr. Lisa Calvacresi, and this is us essentially working on getting this online questionnaire up and going. So what are the goals of the study? We want to try specifically for low-grade glioma to get a little better handle on what is a prognostic model. So if someone comes to clinic, what's prognosis, what are the outcomes, can we guide treatment selection a little bit better? We also want to see if there are genetic variants specific to low-grade glioma. We'd also like to standardize the information that's given to low-grade glioma patients a little bit more. So one of the things that we have noted is, especially when people come from an outside location, they've not been given information on prognosis, on issues relating to family planning. So for example, if there's a young lady and she wishes to consider having a family, what information do we have on becoming pregnant when you have a diagnosis of glioma? How about talking about driving? Do people know that there are rules, generally varying by state, about the Department of Motor Vehicle relating to driving if you have seizure or any other cognitive disability? We also want to look, are there ways that we can make things better? So are there associations between physical activity, exercise, diet, quality of life? And so we're hoping to use some of the mechanisms on the iPhone, like Research Kit and the Health app, to gain information on that. And we're trying to get a feel for what people are willing to do and how much information they might be willing to share with us. So we'll model the registry after a similar registry that we've done for acoustic neuroma. And so if anyone's interested in seeing what it will look like, the Acoustic Neuroma website is hosting that registry, and we're hoping that soon this summer we'll have it up for our low-grade glioma group. And so what are the things that we're going to ask people for? So we'll ask you to confirm your eligibility, and that essentially is to get a pathology report and send it to us so we can make sure that the diagnosis is uniform across the group and that we're actually looking at what we think we're looking at. We will ask you to give us a saliva specimen. So these are some of the little containers, and we would send that out to you. It's quite easy to do. You essentially, it's not glamorous, but it works quite well. You spit into the little well and put a cap on and then send it back to us. Allow review of your medical record, and this gives us information on the different pathology, on genetics that might already been done on your tumor, what sort of treatment you received, and then a little bit of an online questionnaire, just getting information on family history or any risk factors that you might have. And again, this would be the sort of kit that we send out. You open it and the little well is in there. Give the saliva sample and then send it back to us, and just through regular mail. So one thing you can help us with, how can you help the study? Tell other patients in whatever mode works for you, be it internet, phone, group. It's something, it's going to be internationally available, and we're hoping that we can get large enough numbers. Our goal is to get a couple of thousand, so that's basically what we're hoping to do. So we also need help because one of the problems is that when we do these large studies, sometimes the genetic variants can be different for different racial groups. So it's important that we get sufficient numbers for different ethnicity, different races, so that we can guide people of all race in terms of what the outcome or prognosis or best treatment choices might be. And sometimes we do ask people to help others because, as you can see, this is a very internet or web-based study. Some people may not have access to that, so we usually work through other patients, patient support groups, patient associations to try and get to other groups and make sure that everybody gets to join us. So we do need your input. We're right now just developing the app as well as the questionnaire. We're working with the LowGlio group. We really need to know what information have you had a hard time getting. So is it just treatment? Is it understanding the genetic information that you get from your pathology report? Things like seizure management, pain management, insurance, driving. What are the things that you want us to ask about and to try to develop information on? We also want to get a feel for how would you be willing or like to receive this information. So do you feel it should always be in person? Are you interested in having internet resources, webinars, Skype, email? What is most helpful and easy for you to access? Also, when do you want to receive the information? Is at time of diagnosis too confusing, or is that when you feel it's most appropriate? Do you want it always available via the internet, or what are other things that you think would be helpful to you? And the other thing that we're trying to get a feel for is, are you willing to use things like a Fitbit, an iPhone? iPhones can, if you put them in your pocket, actually measure through GPS where you are, number of steps, heart rate, so we can get a lot of information on patients simply by having people put their phone in their hip pocket and walking around. So are you willing to give us access to that? Are you interested in that sort of thing? Would like to try to look at what is the amount of physical activity that people are willing to undertake, and how does that relate to outcome? So glioma at yale.edu, any thoughts or comments that you have, would love to hear about them. Or you can call 617-732-4092, and just wanted to thank everybody and take any questions if people have some.
Great. Thank you so much, Dr. Claus. That was a very, very informative presentation. And everyone now will take questions. So as a reminder, if you have a question that you would like to ask, please type and submit it using the question box in the webinar control panel on the right-hand side of the screen. Dr. Claus, there was one question here about contributing. Is it possible to still contribute to your data registry if the diagnosis was from over 20 years ago?
Absolutely. So that's a great question. So in terms of eligibility, it's persons who are 21 years of age and older, and you can be diagnosed anytime, any place, anywhere. And in fact, it's very helpful for us to have individuals that have variable amounts of follow-up time, because that gives us a much more complete picture of what's happening for low-grade glioma. So great question.
Great, thank you. And can you also speak a little bit about rescue therapy in relation to improve genetic understanding?
So, I don't think, especially for low-grade glioma, we know a lot about that. We're still just looking at what should the initial treatment be. So at least at this point in time, I don't think there's much information on it, which again is why we need to get the whole time span of low-grade glioma.
Thank you. There's also a question about prevention to higher grades. Do you have any recommendation or information about preventative treatments during a stable period during the disease?
Right. So at present, there are no known preventive treatments. And I think that, one of the things that we're trying to do, and there's another consortium that we're involved with called GLASS, G-L-A-S-S, where we will try to collect over time all the samples that a patient might have and to see, can we predict what genetic changes might occur, and that might help us to say, is there any sort of prevention? But at least at this point, we don't know of any.
Thank you. And for the study as well, are you looking for mixed grade diagnoses or only grade 2?
So at least at this point, we are looking for grade 2 only because there's so little study of that group specifically. So we're trying to enrich the study for grade 2 lesions so that we have a large enough sample size to have enough statistical power to address it for that group.
Thank you. And can you also, there's a question here, are there associations of any type regarding the molecular markers to outcomes of immunotherapies?
So that is still a new field. I mean, they certainly are interested in looking at that. There are no trial results at this point, but certainly that's of great interest. And also looking at any sort of markers within the blood with respect to immune factor. So one piece that we're looking at for this low-grade registry is for individuals that are willing to give blood. There are a number of researchers that do want to try to look at what is a patient's immunologic status and how does that affect outcome and response to treatment. So that's another big area that people are very interested in.
Thank you. And can a low-grade glioma on the thalamus be biopsied?
So without seeing the specific case, it would be hard to say. It certainly is possible in general. You know, there are variable thoughts about whether that is a safe thing to do or not. It really would depend on the specifics of the case, but certainly in some instances that is possible to do.
Thank you so much. And what is the standardized counseling given to women with low-grade glioma who want to get pregnant?
So that is an emerging field, and there is some new data that's been looked at by both the MD Anderson group as well as Memorial Sloan Kettering. It's very small numbers because most centers don't have very many women who go on to have a child and have a low-grade glioma. There's anecdotal data that it can increase the risk of progression. I think we've all had a few patients where we have seen that. Most of the larger centers now will have you visit specifically with a counselor to go over the risks and benefits of that and to show the data. So I do think it's a very important conversation, both for males and females to have to understand what the risks and benefits might be. I know at our center at Dana-Farber, patients receive a separate counseling session to go over that sort of information.
Thank you. And do you know if there will be literature available on the registry study that participants can provide, that we can provide to them or to the healthcare providers' patients when it's available?
Absolutely. So we're going to try to be quite good about providing materials both in print, online, and in all sorts of ways so that people can get access to it. So absolutely.
Great, thank you so much. Would a targeted therapy inhibitor treatment be recommended for patients with IDH1 or 2 mutation?
So you'd really have to know the specifics of not only the IDH status, but the specifics of other markers. It would certainly be something that a conversation should be had about, but the further details of the pathology and other markers would have to be considered.
Thank you. And do you have any information on TeRT mutation? If you weren't tested for that at the time of surgery, is that something that you?
Oh, the TeRT mutation?
Yes.
Yeah, that has only more recently been tested for. So I know that if I were to look back at my patients, you know, from 5 or 10 years ago, we did not test anyone for TeRT simply because it's only been a fairly recent finding. So you certainly could have your materials tested for it. Usually materials are saved at time of surgery, so it certainly is possible to go back. I know where we are now, and I think most of the big centers now are adding or have already added TeRT to the panel of things that are tested for. But it would not have been something a few years back, even a year or two, that most places would have tested for. Things are changing fast.
Great, thank you. And with GBM being more prevalent and more aggressive, and oftentimes getting the bulk of the share of research support, what do you feel is the likelihood of any new therapies for GBMs translating relatively quickly into improved outcomes for low-grade glioma patients?
So we do, I mean, that's a great question. When we look at the genetics of both the germline genetics as well as the tumor genetics. Although they are quite different, we do see that some of the low-grade gliomas are moving to become glioblastoma. And so one of the things that we're doing is to look in the low-grade gliomas to try and identify, because clinically they may not look any different, but based on their genetic makeup, to pick out those that are more likely to be high risk and to move on to glioblastoma, so that up front they might be treated more aggressively, whereas individuals with low grade that don't have high risk profiles might be able to spare treatment or wait a longer time until treatment is given. So that's one of the big goals of this group's efforts, is can we tease out right at the beginning who needs treatment and who doesn't need treatment, or at what time might they benefit from treatment, to spare people that don't need aggressive treatment up front, the side effects of that.
Thank you. And one question from another healthcare professional who has had female clients that were diagnosed shortly post-pregnancy. Do you know of any links between the influence of hormones on diagnosis?
Yes, I mean, it's a great question when we're trying to think about what is it that might make a glioma move more quickly during a pregnancy. And the two main thoughts, although this really is not known at all, is obviously hormonal influences, but whether it's also something to do with the changes in the immune system, changes in vascularity, whether that also changes the vascular structure in the brain. So it's not known. You know, those are kind of the three hypotheses that people are talking about, but obviously something important for us to collect more data on, because really each center has a few cases, so we're hoping this registry can also start to address that and also bring attention to the fact that this might be an issue and it's data that patients should have up front.
Thank you. And with regards to low-grade glioma treatment options, is there a consensus on how long Temodar should be used for low-grade glioma?
I would say in general there's little consensus about anything in low-grade glioma. It's probably, and if you look through the literature, you'll see frequently it is one of the most controversial areas in terms of neuro-oncology. So it's because of that issue that we really want to try to focus specifically on it. There's controversies regarding everything, you know, surgery, treatment, when, how much. It's a very tough field. It needs a lot more information.
Thank you. And for those who want to participate but are located outside of the continent, is there any way that they may be able to still participate?
Absolutely. It's open to everybody.
Great. We'll just take a...
And that's part of what, you know, we're hoping the internet can, and that's why we've partnered also with some of these groups like the ABTA, which is based in overseas in Europe. We want to make this open to everybody. Probably at worst, people might have to pay some postage to get the sample back to us, but otherwise it's open to everybody.
Thank you so much. And is there anything else that you would like to add?
No, I just want to thank everyone for listening today, and thanks to the ABTA for supporting us.
Great, thank you so much, and thank you everyone for all of your questions. We truly appreciate you all joining us, and thank you once again, Dr. Claus, for your wonderful webinar presentation. For more information on brain tumors, to help patients and caregivers process the diagnosis, understand a new and difficult vocabulary, and access resources to help make informed decisions, call the ABTA Care Line at 800-886-2282. Let's pause for just a moment to conclude our webinar recording.